For this series of lectures, we're going to talk a little bit about human origins and human migrations, particularly those related to how humans populated the world. And then we'll talk at the end about a bit about some recent migrations that have occurred and how this relates to gene flow and structuring, as we've talked about in the population genetics. First, I want to talk about the human origins as we know them today, and this probably will change on the next time I teach this. But at least at this moment, what we know about human origins. And from that, I want to talk about Neanderthals and the Denisovans and how those interplay, particularly from a genetic standpoint, into our system. So let's start with those. Now, this will take a bit of time, so this will be a little bit of a longer lecture here to get all of this, but we'll get it in. All right, so hominids, humans, Homo hominids have been around for at least 2 million years. And we go back much further as the proto-hominids, Australopithecines and others going back. When I took anthropology in college many years ago, you know, this matrix would have been made up of about 4 different individuals. There was Neanderthals, there were Homo sapiens, of course, Homo erectus, and maybe Homo habilis, and that was about it. But we certainly have had this time. Now, one of the important things to notice about this, besides the fact that, you know, we do have the links back, is that there's a lot of overlap here. And you can see at one point about 2 million years ago, these are millions down here, so that's 2 million to 2000. So, okay, because we're obviously talking about the Paleolithic and the Pleistocene. But we see a lot of overlap, right? And very often overlap for long periods of time. Homo habilis was around for at least 1 million years, and it overlaps significantly with some of the various forms of Australopithecines, as well as some other groups. And then we have this big group, Homo erectus, which was around for almost 2 million years. It only was in recent times, if you think about evolution, maybe in the last 100,000 years that this species disappeared. So we see a lot of overlap. We see a lot of groups that were there at the same time, and they weren't necessarily in the same geographic regions, although many of them were. And there was a lot of course, displacement that went on throughout this time. Okay, so what do we have? Well, let's go back to the Australopithecines, and you can see that there were multiple branches, and probably many more than these will eventually be discovered. But our branch comes along through Homo habilis, eventually up through a group that splintered off into Homo erectus, as well as Homo Neanderthalensis up here, and eventually Homo sapiens that had come off up here. And we had Homo erectus, this group that was here was very widespread. They came out of Africa about 1 million years ago. They spread out through Southern Europe, and around through various areas of Eurasia and into Asia. And they were displaced by a couple of different waves. One of those being Neanderthalensis, which came in, that's the part you see here that's slightly darker, sort of a yellow, I guess. And then eventually the red about 200,000 years ago, and you know, the time frame is a little different there, but time frame, you know, came out of Africa. We had the march of Homo sapiens come out. And displaced many of these other groups over time. So, about 200,000 years ago, we had the development of Homo sapiens. 100,000 years ago or a little more, they moved out of Africa, being able to cross over. So once these areas became wetter, and we ended up with invasions of European areas as well as other areas. And ultimately, that group was able to move over to North America, as well as the entire Pacific area. Now, this movement over to North America, we're going to talk more about that later when the population of the Americas. Now, one of the things that we find out if we go looking at these groups is that somewhere between 2 to 5%, depending on what part of the world you're from, of the modern human genome, so Homo sapiens, is actually Neanderthal. And this is true for almost all the areas that are not found in core Africa. If you go and you look at peoples that have been in Europe for long periods of time, or in Asia or somewhere, you will find Neanderthal DNA. You'll find Neanderthal DNA in Africa as well. It's just not as in the highest number. Okay, so this suggests that, and we saw that overlap, right? So it suggests that we've had some period of time with gene exchange, and it's not just with the Neanderthals, okay, but somewhere along the line, this Heidelbergensis, can't talk, broke off, and the modern lineage of Homo sapiens continued, but these Neanderthals weren't the only other group out there that were widespread. The dissonants, which were described for a cave that was found in Siberia, this cave up in here, were a group, and that they were discovered about 2010. So these are not, or at least they were named in 2010. And it's pretty obvious that they were a major group that interacted both with Neanderthals as well as Homo sapiens, and they heavily occupied all of the Eurasian region and the Asian region, and were responsible for the primary hominid groups that were found out in this region for many years before Homo sapiens came over and through the system. Okay. Now, with these Dissonians, there's two groups. Okay, there was a split. Okay, you can see the split here in the group from Neanderthals, and it's shown here as also as a split. And it turns out that those splits were part of two different invasions of our genomes by the Devonian DNA. So we have not only Neanderthal DNA, but we also have two versions of this other group's DNA that's found here. Now, just looking at one example is that mitochondrial DNA, about 16.5 kb, so 16,500 base pairs, approximately, a little more. The differences between us and Neanderthal is only about 202 bases. Okay, very, very similar. And the Dissonians, it's about twice that, so almost 400, 383 base pair differences. Some of these are just differences, small new differences. Others are actually the size differences that we see. But what we have seen is that there's clearly been gene exchange. Okay, been significant amount of gene exchange between Neanderthals and Homo sapiens. And there has been gene exchange between Neanderthals and the Dissonians, as well as exchange between those individuals and Homo sapiens. So once Homo sapiens came out of Africa, okay, so they were here in Africa, and they moved down and they started to populate theirs, there was quite a bit of obvious inbreeding or interbreeding that went on between these groups. So, you know, at one point there was thought that Homo sapiens were just highly aggressive, came out of Africa and just basically beat up everybody else in some way, either in a Darwinian sense in that they outcompeted them for resources or for reproduction or whatever, or that they physically were more aggressive and warlike or tribal or something. But it looks like now that probably it was more just outbreeding them, you know, and having gene flow in between the groups, potentially with cultural things. They probably may have been able to be tool users and other things more efficiently, and may have even had language abilities that were higher levels interactions, but they basically were able to beat out the other groups. But even with that, there was gene exchange that went on. Okay? And if we look, when you mix genomes, it doesn't matter whether you mix populations, you know, that have been isolated for a while, or you mix, in this case, species together. That's called admixture. Okay, and so we have an admixed genome. We have this modern human genome. Okay, that's the solid lines into here, and we have this Neanderthals DNA. That's again somewhere between 3 and 5% on average. You know, you can have 0 depending on the things that go on with genetics, assortments. But we have a bit of that, and we have some of this Dissonian DNA, this admixture. And it just depends on where you are. Now, the biggest portion of them, because this species of hominid was primarily in Asia and Eurasia, is that's where the greatest concentration is of this exchange of DNA. And it was somewhere between 4 and 6% of the DNA there. So equivalent into what you see in Western Europe as far as the Neanderthal DNA. So we're a mix. We're a mix of, we have combined DNA from various groups. Now, this dissonant, I keep saying it the right way all the time, is going to be important because when we talk about population of the Americas, it turns out that this DNA is actually some DNA that shows up here, okay? And it's in a way that is unusual. We'll talk about that a little bit later. Okay? All right, so what about some of this? Well, let's think about some of the genes. So what I want to do is kind of look at the differences and look at the similarities, right? Well, one of the places that we know there's some differences is in the toll-like receptor genes. And these are genes that are on cell surface, and they help us both detect and defend ourselves from fungi and bacteria and all kinds of things, okay? And there's a number, there's a series of them found around, but one of a group is found on chromosome 4. And these toll genes are, you know, fairly normal systems. They're found on the surface, so they have an extracellular part, right? So that's what the red is here, saying that this part codes for a part of the protein that's going to end up outside of the cellular membrane. And then you've got a cytoplasmic region. So you got an anchor region that's inside, and you got a TM that's transmembrane. So they would literally be, here's the cell surface, and this would be a protein that's sticking out somewhere out here, but it also has an internal part that's cytoplasmic, okay? Now, if we look at the frequency of these genes, okay, there's a Neanderthal-like form, a mutated form of it that is out there. And if you look at the presence of this, you can see where it's highly prevalent, okay? It's found, this gene's found, I said there was some in Africa. There's various forms of it found in Europe. And then there's a large populations of it are found down in Asia and down in the Pacific areas. And it's also found, and this is going to be very important later on, it's found off in South America, okay? Not a huge proportion of it in native North Americans, and this is from peoples who are native to these regions or have lived there for long periods of time. One of the cool things about it is, you know, these are proteins that protect us, right? Because they're cell surface proteins. These are immunity genes. That's one of the things it's helped with. And the other is it made us susceptible to allergies, okay? Because we respond. It triggers the typical responses. So it's really thought to be one of the gene things that we picked up from our Neanderthal buddies that actually work in some way to protect us, but also cause our systems to overly respond. And there's been some of the COVID discussions about whether the Neanderthal genes that we have actually make us more susceptible to COVID or less susceptible. It's I think some of those data will be popping up in the next year or so, and maybe a few years, and it'll be kind of interesting. Okay. All right, well, here's a real map of it, and we sort of, these are chromosomes. So that's the X chromosome. There's 22. So chromosome 1, 2, 3, 4. You know, and they're kind of structured by their relative size. Obviously, chromosome 1 and 2 are large chromosomes. Chromosomes 12, 13, 14, 15 are medium size, etc. So what we see here is that the gray part that goes through here is modern human. And then the colored parts represent the gene regions that are basically ancestral, okay? They're things, they're not saying we got them from Neanderthals, but our zones is saying that they're what we think are the same. And you know, you think about that because as species come from a common source, they maintain some of those genes. So this isn't saying that every blue spot here we got by inbreeding or interbreeding, is that it's saying that that's the things we carried with us from our past. We both carried those, okay? Modern humans carried them as well. Now there may be some that exchange, but for the most part, it just means that we have a common ancestor, which we do, right? And if you go back and you figure out what that common ancestor is, you carry those genes with you. So as you can see, there's a lot of similarity there. We carried with a lot of genes from these populations, or brought them in, okay? And from Neanderthals, we've either brought in or gained from their past, one way or the other, either interbreeding or from having a common ancestor. We've carried forth a lot of genes. And there's a bunch of different things. One is one that apparently helps that helps people from Tibet survive at low oxygen temperature. You know, the Sherpas that can go up Everest and things without oxygen until they get to extremely high altitudes. There's some that are found helps respond to radiation. Immune system, again, that's the toll genes that are found here, all of this region. On chromosome 4, there's some immune response ones that are found in different individuals. There's all sorts of things, freckling and skin pigments that's found, in humans that probably that are European Homo sapiens that probably came from Neanderthals. These aren't found in Asians as much, and it's kind of an interesting one. So anyway, nice pattern. So we've either kept genes, okay, in one form, or we have brought genes together from other combinations through our intermixing and admixture is the word of different species combinations, near species or subspecies combinations. Okay? Now, one of the cool things they've done in recent time is they've looked at the methylation patterns. Okay, so remember, methylation is very important for us for epigenetic and for protein structure. And they've seen that there is some, if you compare present day, which is along the bottom here, and then you compare that with the other forms, make a matrix here, so X, Y coordinate. We see a lot of methylation differences here, okay? And what it's basically saying is that there is a generalized common pattern, which you would expect, okay, but it's kind of cool. Although there are differences, I mean, you can see here that the Neanderthals have a little here, and that was a much wider band that was found here in the dissonants. But if you look at the red part right there and the red part right there, that's methylated region that's found in modern. So it's still the same methylated region that was there, okay? And then, hypomethylated, or the greens, and you see some of the same things. Again, this is just one little gene region that just popped up here. It's not trying to say the whole genome. Okay? Well, why is that important? Well, remember the Hox genes, those genes that we talked about, this whole complex of genes that are involved in the timing and formation of fetal development and body parts, and that we have Hox genes that control neural structures and our arms and legs and all that different parts. Okay. Well, there was a study done looking at just sort of the Hox genes themselves and looking at the methylation patterns for that. Okay. And it looks like that early hominids, okay, early humans, were hypermethylated. And that, I mean, you can see if you look at all the methylation levels here, and you look down here in present day, we don't see that. We've lost a lot of that methylation pattern that we went on. We tend to be, hypomethylated in some of the Hox genes in the same areas. It's kind of interesting that there was this real difference between the two in the methylation forms. Now, from that, okay, because these are Hox genes, and you think about development in systems, a really cool study was done where they would look at these levels, okay, of methylation change. They would look at the promoter, okay, which is, obviously that's one of the big methylation sites that controlling things. They'd see a lot more methylation in proto-humans. They would see that this caused downregulation, okay, or loss of function. They were able to think about this in form of how the body works, and going through this and looking through all the genes, or a whole series of genes that they could work with, that particularly the Hox genes and others, they were to say, okay, we know what Neanderthals look like. Okay, we've got the bone structure, the craniums, we've got things we know they were robust hominids. We know what chimps have and forms, obviously, because chimps still exist, so we can look at their morphology. We really don't know what the dissonants look like, because there's only really tiny fragments from that have never been found, jaw fragments and other things that were used to describe them, and they're clearly different, partial skulls, but there's really no. So what they were able to do is they would take this and compare it against known systems. So they said, okay, well, this gene must be active this gene is downregulated, okay? Well, what does it mean? Well, it's downregulated here in the chimp, it's upregulated here. So, and in modern humans, so that must mean X or Y, right? And if you kept playing with that and running a bunch of fancy computer simulations, they were able to, say, okay, these things should be similar, these things should be different. And if they're different, which direction should they be different? Should they be smaller? Should they be larger? Whatever. And ultimately, they developed a profile of what these individuals should look like. Once we find a full skeleton or a full thing, we can kind of see what they might look like. And in 2019, there was a full anatomy description brought out. And I think that's one of the cool things, based primarily on these methylation maps. Okay, looking at all the places that you could have it, and they would predict the trait would either be increased or decreased in expression. Like pelvic size was predicted to be decreased in size. And then these are some of the things that they would end up looking for, okay? The flat cranial vault, so low forehead, they would have, wouldn't have enlarged molars, okay? Pulp of the molars. These are all just genes that we know that respond in humans, okay? In chimps and Neanderthal differences, just sitting there with a computer comparing them and saying, okay, this gene would be hyperexpressed, this gene would be normally expressed, this one would be downregulated. And so we predict, well, not me, but the prediction is that you'd have these kind of structures. Broad ribs, okay? They would have, wide fingertips. It's just, anyway, I thought that was just an amazing outcome when I read this paper in Cell about a year ago. I said, holy cow, it's pretty impressive that you could take this methylation, sit there and basically reconstruct this entire individual for the most part that you have no way of ever seeing, and it's really cool. But it's done again for methylation and knowing, because of epigenetics and understanding that, what genes are going to be upregulated and what genes are going to be downregulated. Okay. All right, well, that's basically our origins, okay, for deep origins, okay. We get the Homo sapiens. Now we've got them, okay. How did we get the world populated with these Homo sapiens? And that'll be in our next lecture. We're going to talk a little bit about populating the world.